Okay, so here is our synapse again, right? I've added more detail, which is just the thing you want, right? So again, here's our presynaptic, membrane, right? Our axon terminal. Here's the sodium rushing down, right? Depolarizing as it goes. Remember, reducing the charge difference between the inside and the out from the resting potential, about -65, more negative inside versus out, to the threshold potential of -55, at which now voltage-gated calcium channels are going to open, right, en masse. And when they open, they allow for calcium to flow, right? They're selective for the flow of calcium. Now, calcium comes from your diet, right? Calcium comes from dairy products. It can be fortified in soy milk products and things like that. Calcium is an important nutrient for neuronal function, right? It turns out that calcium is going to be found in high concentration, right, in the extracellular space. The membrane is pretty impermeable to calcium until you've reached the threshold of -55, and suddenly all these protein holes open up that allow for calcium to go through. So calcium enters. It's driven by our concentration gradient force, right, because it's all out here, very little inside. And it's also happening, this opening of these channels at the threshold of -55. So it's still negative on the inside. The electrical force also contributes to the movement of calcium inside the axon terminal. When it comes in, it interacts with, that's all I'm going to say, it interacts with components, ultimately, of this docking complex, okay? So calcium comes in and it's going to influence, the structure of this docking complex in such a way that a piece of the phospholipid bilayer membrane on the vesicle is going to be pinched off and made continuous with a piece, with the membrane, of the axon terminal, the presynaptic membrane. So you're going to suddenly have a hole, from the inside of the vesicle, out into the great big, synaptic gap or synaptic cleft. And remember our force that we'd sort of stored in here. We have vesicles that are filled with neurotransmitter, high concentrations. Suddenly, there's a hole from this highly concentrated, assembly of, neurotransmitter here into the big, open synaptic cleft. The neurotransmitter comes streaming out, okay? It exits this cell, right? This process by which the neurotransmitter leaves the vesicle via these pores that are formed, upon calcium entry is known as which means exiting, cytosis, which is the cell. So this is exocytosis, the release of neurotransmitter into the synaptic cleft. Now, what I want you to consider is that this doesn't happen all the time, right? This cell is not just leaking neurotransmitter on a regular basis. We do have cells in our body that, release chemicals on a regular basis. They're called gland cells, right, on a more regular basis. The release of neurotransmitter here, of chemical here, is contingent upon the arrival of an action potential. It only happens when, the neurotransmitter, the sodium current arrives at the axon terminal. This is an actual translation process that's occurring here. We have a message, an electrical message in a neuron, right? The sodium current that's rushing down, okay? Which, occurred only when you depolarize the threshold somewhere else in the cell. And when it arrives at this, axon terminal, its arrival is coupled to the release of neurotransmitter. This is a specific message now that's being sent, based upon the arrival of an action potential. Now this neurotransmitter will diffuse, right? It'll distribute itself across the synaptic cleft, and some of it will reach the postsynaptic membrane and interact with specific binding sites that you find on these various receptors that are embedded, in the membrane of the postsynaptic cell. So let's go over our, set here, our sequence here. The action potential, the sodium current, arrives, right, at the axon terminal. It depolarizes the terminal to the calcium channel threshold, which is about -55 millivolts. Calcium will rush in, right, driven by the electrical force and the concentration gradient force. The vesicles will then fuse with the membrane, and you'll have exocytosis, the release of neurotransmitter into the cleft.